Assalamu alaikum students. Before going to our educational topic, please subscribe my channel Muntzir Mehdi Science TV on YouTube. Press the like button and share this lecture to your friends and classmates who have no access to it to add in their science education. And those students that are preparing privately can benefit from our channel very effectively. Let's come back to our topic, that is Kreb cycle. It is known as after the scientist named Hans Krebs, who discovered it in 1937 and received Nobel Prize for Physiology and Medicine in 1953. So the Kreb cycle name is derived from its discoverer name Sir Hans Kreb. This cycle is also named as Citric Acid Cycle because the first product of the cycle is citrate that plays a central and key role in citric acid cycle. The citric acid is also known as citrate. Kreb cycle is also named as TCA cycle, that stands for tricarboxylic acid, because it consists of three intermediates like citrate, aconitate, and isocitrate. Each possesses three carboxyl groups. This cycle is known by these three names. The Kreb cycle is a key metabolic pathway that connects carbohydrates, proteins, and fat metabolism. It is a central pathway into which many metabolites pass. It consists of many reactants that generate NADH and FADH, which are in turn utilized to produce ATPs, the energy currency of the cell. The Kreb cycle possesses a series of chemical reactions that provide stored energy of organic compounds via oxidation process. In addition to supplying energy, Kreb cycle is also very important because it provides many raw materials that are used in different parts of the body. These raw materials may be amino acids or NADH. An important fact about Kreb cycle is that it was one of the earliest established components of the cellular metabolism and may have originated abiogenically, which means that it originated even before the life started. Abiogenesis concept is that life originated from non-living things. As Kreb cycle is a central and main metabolic pathway. So what do you understand by central metabolic pathway? Let's imagine that when we eat food, it is digested, absorbed, and utilized, and is first processed by initial metabolic pathways. These initial metabolic pathways provide some common products that are used by the central pathways like the Kreb cycle. Kreb cycle to synthesize energy. As if we take the example of carbohydrates that are split into simple sugars, glucose. This glucose is first and initially processed by initial metabolic pathways like the glycolysis. It produces pyruvate, and similar products are produced by the initial metabolic pathways of fats and proteins. This pyruvate is then taken into central metabolic pathway of Kreb cycle. So it provides a central metabolic pathway that connects the proteins, the carbohydrates, and the fat metabolism. Now an important question emerges in our mind, that how does the Kreb cycle provide energy? To know the answer of this question, consider the metabolism of carbohydrates again. As we know that we obtain simple sugars at the end of the carbohydrate digestion like glucose, okay, and this glucose undergoes all the metabolic pathways like glycolysis, Kreb cycle, and electron transport chain. So 1 mole of glucose provides approximately 36 ATPs. But where does this energy come from? The single body. This question can be answered by the chemical process of oxidation. Let's see what happens when through glycolysis and an additional reaction we break down glucose to small 2-carbon compound called acetate, that is then enters the Kreb cycle. If we consider Kreb cycle, which consists of 10 intermediate molecules that start and end with citrate. When we add acetate to Kreb cycle, that is a 2-carbon compound, this cycle takes a complete turn and liberates 2 molecules of carbon dioxide as a waste product, which means that the two carbons that we added in the form of acetate are liberated in the form of carbon dioxide. This carbon dioxide is produced due to the oxidation process that occurs in the Kreb cycle. As a result of different enzymes, electrons are taken into NADine to make it NADH and FAD to make it FADH. These electrons are then subsequently used during electron transport chain that we will discuss in our next video to make ATP through ionic gradients. All these informations may seem a bit difficult, a bit disturbing, but these are very important informations and knowledge for understanding the Kreb cycle. That is a very important topic for annual examination like glycolysis. You can also watch glycolysis lecture by subscribing our channel. Now we see how does Kreb cycle take place in the eukaryotic cells. In eukaryotic cells, it takes place in the matrix of mitochondria. On the other hand, in prokaryotic cells, like bacteria that possesses no mitochondria, Kreb cycle occurs in the cytosol. There is a very important reason why Kreb cycle occurs in the center of the mitochondrial matrix. This is because of the presence of the electron transport chain in the membrane of the mitochondria. This electron transport chain of various enzymes that utilizes the products formed in the Kreb cycle to produce energy in the form of ATP. So all these are the important descriptions that occur before the main cycle. So when the end product of glycolysis moves from cytoplasm towards the mitochondria, after the series of modifications, as soon as pyruvate moves into the mitochondria, we can begin next steps that is known as Kreb cycle. Kreb cycle begins with acetyl CoA. But where does this comes from and what is its role in the Kreb cycle? This is very important to understand. Now one of the important biochemical pathway that runs before the Kreb cycle is glycolysis. During glycolysis, we supply glucose, a 6-carbon compound, into 2 molecules of pyruvate, a 3-carbon compound. What happens next is that this pyruvate is acted upon by a set of enzymes collectively known as Pyruvate Dehydrogenase complex. In this reaction, coenzyme A is also involved, which is a coenzyme derived from vitamin B5. In this reaction, 2 carbons are transferred from pyruvate to coenzyme A to synthesize acetyl CoA. One additional carbon present in the pyruvate is lost in the form of carbon dioxide. In this reaction, one molecule of NADH is also produced. So this is how acetyl CoA is produced from pyruvate. This acetyl CoA is a 2-carbon compound and is the starting point of Krebs cycle. In this molecule, the coenzyme A, the part which is derived from vitamin B5, acts just like a carrier, which involves its 2 carbons, combines it to the Krebs cycle. So during the first step of the Krebs cycle, acetyl CoA combines with the oxaloacetate. As I told you, acetyl CoA, a 2-carbon compound, combines with a 4-carbon compound oxaloacetate. This combination leads to the formation of citrate, that is a 6-carbon compound formed by the combination of 2 carbons of acetyl CoA and 4 carbons of oxaloacetate. This reaction is catalyzed by the enzyme citrate synthase, which is an allosteric protein that is activated by AMP and NAD and inactivated by ATP and NADH. The function of the citrate synthase is self-explanatory by its name. For what it does, the citrate then undergoes a series of enzyme-catalyzed reactions during which it loses 2 carbons of acetyl CoA in the form of 2 molecules of carbon dioxide. The conversions, which involve up to 10 chemical reactions, are all carried out by enzymes which utilized energy obtained through oxidation to combine electrons to NAD and FAD. Let's study what takes place next with the citrate. During the next reaction, citrate is converted into its isomer known as isocitrate. This second reaction comprises of two phases. Understand that aconitase is that particular enzyme that converts citrate to isocitrate through an intermediate unstable acid aconitate. This reaction takes place in two phases. Aconitase enzyme during first phase acts as a dehydrase which loses one H2O molecule from the citrate and converts it to aconitate. Same enzyme aconitase during the second phase acts as a hydrase, which means that it adds a water molecule to aconitate to generate isocitrate. We can also say that during first phase, citrate is converted to an acidic form aconitate by the loss of water, that is showing dehydrase process, whereas during the second phase, aconitase enzyme converts aconitate to isocitrate by the addition of water, is showing hydrase process. Both these reactions which I have just now shown by a single arrow are catalyzed by the enzyme aconitase, that is an iron containing enzyme which reorganizes the citrate atoms to form its isomer isocitrate. Now you may question what was the need of to convert citrate to isocitrate. Why cannot the citrate just move forward with the remaining Kreb cycle? Well, its answer is that the arrangement of atoms in the isocitrate is more suitable for the upcoming few oxidation reduction reactions that are about to happen during the Kreb cycle. So during the isomerization of citrate to isocitrate, an intermediate is also obtained which is known as cis aconitate, an acidic form as I have already told you, but there is no need to go into the further details about it. During the next step, isocitrate is converted into alpha ketoglutarate with the help of an enzyme Isocitrate Dehydrogenase. Now we look at the first of the four oxidation reduction reactions of the TCA cycle. These oxidation reduction reactions are those which produce energy in citric acid cycle, and all of these four reactions utilize enzymes which are known as dehydrogenases. Now what do these dehydrogenases enzymes do in them, and how do they generate energy? That is very important for you to understand to analyze their methods. See, if you want to understand the term dehydrogenase itself, it literally means removal of hydrogen. So dehydrogenases are basically those enzymes that remove hydrogen from a molecule. As you can memorize, the removal of hydrogen is also known as oxidation, and the hydrogen that is lost from one molecule, removed from one molecule, must be passed on to the some other molecule. The second molecule that receives the hydrogen gets reduced in turn. So this is the basic mechanism of the dehydrogenase enzyme's function. They taken out hydrogen group from one molecule, oxidizing it, in turn attaching that hydrogen atom to another molecule, carrying on its reduction. That is why the reactions carried out by dehydrogenase are known as oxidation reduction reactions. Let's again we consider our citric acid cycle. We were discussing that point where isocitrate will now be acted upon by the enzyme isocitrate dehydrogenase. So now understand what happens in this reaction. That is actually a two-step reaction during which the enzyme isocitrate dehydrogenase first removes hydrogen from isocitrate, thus oxidizing it. That removed hydrogen is attached to NAD to form it NADH, thus accomplishing the process of oxidation reduction reaction. But there is an additional step that takes place in next, which is catalyzed by the same enzyme isocitrate dehydrogenase. A carbon atom is also removed from isocitrate in the form of carbon dioxide molecule, which leads to the formation of a 5-carbon compound alpha ketoglutarate. This complete biochemical process that just happened here, known as the oxidative decarboxylation. Oxidative because oxidation of isocitrate takes place, during which a hydrogen is taken out from isocitrate, and decarboxylation as a carbon group is also removed from isocitrate. The conversion of isocitrate to alpha ketoglutarate is followed by the second oxidative decarboxylation reaction. The alpha ketoglutarate, a 5-carbon compound, is then converted into Succinyl CoA in the presence of an enzyme alpha ketoglutarate dehydrogenase complex. Alpha ketoglutarate dehydrogenase is a complex enzyme, but there is no need to discuss further it, that catalyzes the similar oxidative decarboxylation of alpha ketoglutarate, during which also a hydrogen gets attached to NAD to form it NADH. As this reaction is also a decarboxylation, so a carbon is removed from alpha ketoglutarate to be lost in the form of carbon dioxide. Coenzyme A is also used in this reaction. Therefore, the product will be a combination of molecule that is known as Succinyl CoA, that is a 4-carbon compound. During the next step of citric acid cycle, the Succinyl CoA is converted into succinate. This reaction is catalyzed by the enzyme Succinyl CoA synthase. Now let's try to understand what happens here in this reaction, as it is very important concept and most of the students did not know, and left this most important point of view, that the number of carbons remain the same in this reaction. Both Succinyl CoA and succinate have 4 carbons each. Now an important question comes in our mind, that when both products have the same number of carbons as 4 carbons in each, then what changes, what converts? The answer is that Succinyl CoA is a high energy compound, and its stored energy is utilized to attach an inorganic phosphate to GDP, that is converted into GTP. GTP is quite similar to ATP and is later used in the synthesizing ATP. The coenzyme A attached to the Succinyl CoA also gets detached in this reaction. So what takes place here essentially? Answer is that the Succinyl CoA uses its high free energy and its coenzyme A to be converted into, to be changed into succinate and a molecule of GTP, which is an example of substrate level phosphorylation, a process of generating energy that we will discuss in our upcoming videos. Otherwise this video become more longer. Now we have succinate, a 4-carbon compound. Actually the number of carbons do not change from here onwards. All the compounds which will occur next in the cycle only have 4 carbons. The last produced compound oxaloacetate also consists of 4 carbons. That oxaloacetate joins with another molecule of acetyl CoA to form citrate of TCA cycle. This citrate will be starting point of upcoming cycle. Now again comes towards the succinate, that is catalyzed by the enzyme Succinate Dehydrogenase. As you can understand by its name that this is a dehydrogenase. So it releases hydrogen from succinate, and this removed hydrogen is taken into flavinadenine dinucleotide, shortly called FAD. In turn, that is converted to FADH, and the succinate is converted to fumarate. FAD is hydrogen acceptor in this reaction, not NAD, because combination of hydrogen to NAD requires more and more energy in this reaction. Therefore, FAD is utilized here because it requires less energy change to get reduced to FADH. Free energy change for 4 carbon compounds is less as compared to 5 or 6 carbon compounds that were present during the first half of the cycle. However, there is an exception to this at the end of the cycle that we will discuss in a short detail. The enzyme for the conversion of succinate to fumarate is Succinate Dehydrogenase, that quite similar to Aconitase is an iron sulfur protein and is also very important because Succinate Dehydrogenase is directly associated with electron transport chain. Next step is a hydration reaction, which means, as you know, we add a water molecule to fumarate and convert it to malate. This reaction is catalyzed by the enzyme fumarase. Nothing special occurs in this reaction more. Let's see next reaction. Here oxidation will be, during which we ultimately oxidize malate to oxaloacetate. This conversion takes place in the presence of an enzyme malate dehydrogenase. The NAD molecule gets reduced to NADH again, which will be used during the electron transport chain. As NAD gets reduced to NADH here, so now you may question why NAD gets reduced here instead of FAD. As I answer you already that the free energy change for the compounds during the end of the cycle is less as compared to the first half of the cycle. Well, this is an additional point of view that I am talking about. As this reaction is present at the end of this cycle, so a molecule of NAD is getting reduced here because this is due to the fact that the oxaloacetate that is being produced here is quickly being utilized by combining it with acetyl CoA to form citrate, and the second produced product NADH is also being used quickly during the electron transport chain. So the standard free energy for this reaction is significantly positive, unlike that for the other steps of the Kreb cycle. So now oxaloacetate again combines with a molecule of acetyl CoA and leads to the formation of citrate with the help of an enzyme citrate synthase. In the presence of an enzyme citrate synthase, an H2O molecule is also taken in this reaction. This accomplishes a full turn of the cycle, and same sequence is repeated again and again and again to get more and more energy for the organism's body. Whereas if we consider the cycle, we can analyze that what we obtain from the cycle. We took 2 carbons to the cycle in the form of a molecule of acetyl CoA. These 2 carbons were discharged in the form of 2 molecules of carbon dioxide here and here. During oxidation reduction reactions, we change 3 molecules of NAD to NADH and 1 molecule of FAD to FADH. We also novitiated 1 molecule of GDP to GTP. Now there are some other important point of views regarding the Kreb cycle that you must identify to understand a complete knowledge about this most important biochemical process. First, we will see the summary reaction of the citric acid cycle. After that, we will see the energetics of the citric acid cycle, which means that how many ATPs are achieved for every 1 molecule of glucose that moves in the Kreb cycle. First, see the summary reaction. So now the net reaction of Kreb cycle is 1 molecule of acetyl CoA, 3 molecules of NAD, 1 molecule of FAD, 1 molecule of GDP along with 1 molecule of inorganic phosphate and 2 molecules of H2O. All these are associated to give 2 molecules of carbon dioxide, 3 molecules of NADH, 1 molecule of FADH, 1 and other molecule of GTP and 1 molecule of coenzyme A. So now what is the jargon? So let me decode this reaction for you. On the upper sides are unprocessed materials that are being utilized during the citric acid cycle, and on the lower sides are the products that are being generated during the citric acid cycle. So in every single turn of the cycle, we utilize 1 molecule of acetyl coenzyme A that combines with oxaloacetate to form citrate. NAD are utilized that are then reduced to 3 molecules of NADH. 1 FAD molecule is also utilized, which is also reduced to 1 molecule of FADH. This FAD NADH is achieved during the conversion of succinate to fumarate. Two molecules of carbon dioxide are achieved during the conversion of pyruvic acid to acetyl CoA and alpha ketoglutarate to succinyl CoA. As I described you recently, one coenzyme A is generated during the alteration of Succinyl CoA to succinate, where succinyl CoA removes the coenzyme A, and one molecule of GDP combines with one molecule of inorganic phosphate and leads to the production of one molecule of GTP. Two water molecules are also absorbed, one in the production of citrate and other in the hydration of fumarate. So now I have talked about the summary reaction of citric acid cycle. Now I will discuss a little about how much energy is produced during the citric acid cycle. Most of the obtained energy is through the reducing equivalence, the NADH and FADH, that are subsequently utilized during the ETC. If you consider the happening of these two biochemical pathways in the mitochondria, they happen very close to each other. Citric acid cycle is happening in the mitochondrial matrix and ETC in the membrane of the mitochondrial matrix. ETC is the short word of electron transport chain. So the NADH and FADH that are being generated by the citric acid cycle, Kreb cycle, are easily utilized by the ETC for the production of ATP. The description of how these ATPs are generated in the electron transport chain and how many ATPs are generated during ETC, we will be talking it about in our next upcoming video, that will be for the topic of electron transport chain. At the end, we will discuss about the energetics of the citric acid cycle. As we have known that during Kreb cycle, 3 NADH and 1 FADH molecules are produced during Kreb cycle as reducing equivalents. It is an important point to keep in mind that 1 NADH molecule equals to 3 ATP molecules, whereas 1 FADH molecule equals to 2 ATP molecules. As we have 3 NADH, 1 NADH will be equal to 3 ATP molecules. So 3 by 3 equals to 9. 1 ATP will be obtained from GTP and 1 FADH molecule will give 2 ATP molecules. So 12 high energy ATP molecules are obtained in one repeat of the citric acid cycle, as 2 pyruvate molecules are obtained from 1 glucose molecule. So citric acid cycle repeats twice. So total number of high energy ATP molecules will be 12 by 2 is equal to 24 ATP molecules. So students, Kreb cycle is very important topic that often comes in annual examinations. You must prepare it very well and would become fond of science knowledge so that you can service your country, your nation very well. It is my encouragement for you in this lecture. So thank you for watching this your video.